Hello, everybody, and welcome to our webinar series, Elmarco Talks. This is where we invite our industrial and academic partners to come and talk about what they're doing with electrospinning, some interesting things. Today we have a special guest with us from Materic, is our event partner. They'll be talking today about tissue engineering on electrospun scaffolds, biopapers, and beyond. So I introduce, we have two speakers today, but one of them's having some technical difficulties. James is. So Dr. Stephen Farias is going to take the helm. So we'll see. I'll introduce myself. I'm Joshua Manasco. I deal with business and application development for Elmarco. I have my colleague here, Katka Rubichkova. She's a sales manager for Elmarco. So I'll first introduce Stephen, and then we will talk a little bit about Elmarco, and then we will get into our talk. So Dr. Stephen Farias, he is the Chief Science Officer from Materic. He has a PhD in material science and engineering and over a decade of experience in research and industry. He has a technical expertise in nanoparticle synthesis, composite fabrication, and electrospinning, as well as materials characterization. And he's led the development and commercialization of multiple novel material solutions from lab to commercial scale. And so one thing I wanted to mention is this is our 8th and final webinar for the series. So we'll be having more webinars coming in the near future, sometime in the autumn. And you'll be receiving some things to give feedback and some topics that we will be presenting in autumn. So with that, I'll hand it over to Katka to talk a little bit about Elmarco.
Mm-hmm. So just a little reminder about Elmarco, about our company. So we are a global supplier of the technology called Nanospider, which is based on the electrospinning method producing nanofibers. We are located in a small country in the middle of Europe, and Elmarco exactly is located in Liberec, which is a little town about 100 kilometers far from Prague, north of Prague. And we have pretty much all facilities are concentrated here in this little town, including assembly, research. We have a department of mechanical engineers where we are designing the machines. We have a big facilities for testing the equipment, which have been tested before they are distributed or transported to customers. From the map, you could see that although we are centralized here in this little town, we are more or less global companies. So we have a sales all over the world, and we are basically supplying the machineries to whoever needs it for the nanofiber production. We are more than 20 years in the market already. We have a lot of patents dedicated to the technology itself, and we have hundreds of machines sold so far. And we are doing good. So this is how the technology look like. So we are not working with nozzles, not with needles. We are working with wire, which is basically the main electrode, the spinning electrode, which is being used for generating the nanofibers. We are working from solutions, so polymer needs to be dissolved to generate the solution, which is brought on the top of the wire, and by applying the energy in form of high voltage power supply, the fibers are generated and are coming towards the collector, where are being collected on the moving substrate, which can be any kind of textile, non-woven textile, or any flexible, let's say, material which is rolled through the electrospinning chamber. So that's how the technology look like. This is briefly the portfolio of our machineries that we are delivering, starting from the really small ones for laboratory research, initial research, going through a little bit bigger one, half-meter size machine, which has been recently upgraded, and I will touch to that a little bit later. And then, of course, our biggest goal is to sell the industrial scale machinery, which are fully equipped with also other peripherals to really fulfill the requirements of the real industrial long-term production. So that's the machine I was talking about, and actually, this is nice to mention, especially today when we're talking about the tissue engineering application and generally in medical application, because this machine has been actually designed for this application. Not only that, we are upgrading it with another electrode. So in nowadays we have 3 electrodes, free spinning electrodes instead of 1 electrode, which is obviously generating more production throughput. But also this machine is a little bit versatile because we are offering 2 different collectors. One is a belt collector, which is here for making either self-standing membrane by continuous rotating of this belt, where you can build up a kind of thick layer of nanofibers on the top of it, or it helps with conductivity of some problematic substrates to be coated. And then we have another type of electrode, which is the rotating electrode to help you to generate the aligned fibers, which is kind of idea for growing the cells. And I think Stephen will talk about that in his presentation. So this entire machine offers really a lot of options, and we hope it will be kind of nice machine to work with, especially in the medical segment. That's the briefly summary of the whole application. Obviously, we are into air filtration. That's 80% or even more. There are other applications which is coming up, and it's a liquid energy. It's a big boom with the battery separators at this moment. We're doing a lot of research in this direction. Performance apparel is the very promising application because of the functionality of the membrane, functional membrane. And medicine, that's the also broad topic, and we are going to touch just one of the part of it today, tissue engineering drug release. That's about Elmarco, and we can hand over the word to Stephen.
Well, I wanted to make one note is that if you have questions, please put it in the Q&A section, and then we'll get to those as Stephen finishes up his talk. So, Stephen, without further ado, I'm going to stop sharing and let you share your screen and tell us a little bit about Materic. And
So I'm excited to give the talk today on tissue engineering on electrospun scaffolds. And we have a line of products ourselves in the space that we call biopapers, but we're also going to talk broadly about a number of academic publications and different applications that we've seen in the space from our own customers and collaborators. And I'll go ahead and get into it with just a quick introduction to Materic. We're focused on manufacturing the materials of the future. That includes specialty textiles and nanomaterials like the electrospun materials we're going to talk about today. That we've done a lot of different work on both the Elmarco NS lab and to translate things onto the commercial tools as well. But also in specialty inks and controlled release, where we might be creating a formulation or an additive to be added to a textile material or non-woven material, or do things with individual clients where they might have, in this case, an active pharmaceutical ingredient, an antimicrobial, or some growth factor additives that they want to add to the textile material, that we can do so in a controlled fashion, which is an important part in the tissue engineering space. And then 3D printing as well. And some of that actually includes bioprinting and inks. And that's actually how we got into what we call our biopapers in the first place. There were some labs out of the Naval Research Lab here in the US that were doing printing of different cellular materials. And that was new to us. And then we were asking, they're clearly printing onto something that looked like a piece of paper but wasn't. We asked what it was, and it was actually electrospun gelatin that they called biopapers. And it was a great media for them to be able to do their bioprinting and tissue engineering off. So that's actually what got us. We were already in the electrospinning space, but really got us into this tissue engineering space was with that set of partners. And we do custom designs and manufacturing of advanced materials, and everything from someone coming to us with a piece of paper, a sketch, an idea, to maybe they have their own prototypes and focused on ways in which we can drive to scaled manufacturing. And so we really like to fill in that gap that's often there between maybe a concept or a benchtop idea that's been in the academic space, and what does it take to get it into that commercial space. We're based in Baltimore. We've been here since 2012, and we actually have some subsidiary companies under our umbrella. You might be familiar with dipole materials, which is kind of where our electrospinning work started. But we've got 30,000 square feet of manufacturing space in our own building that we control now, and that allows us to both do benchtop development as well as scaled piloting and eventual manufacturing for our clients. Um, and there's lots of different parts of that space relevant to this tissue engineering topic. We've got electrospinning, we've got different textile treatment space to do that formulation or post-processing that we need, different imaging and metrology techniques, including IRCTs that can look into tissues, and some biolab space to be able to run into that. But specific to why we're here, really it's because we recognized a technology bottleneck that all these different ideas and new technologies get generated in the space, but very few actually get shaken down into the production space and eventual industrial products. And just to give you a number, in the US until 2022, the government spent $90 billion on academic research, right? So that's going specifically to basically funding PhD and research programs at academic institutions. That does not include any of the private money going into research or things that are funded by like specifically industrial research and development. Of that $90 billion, so in 22, there are only about 850 products actually being generated from those research institutes. That means that we're spending about $100 million of research money within the US per product that actually comes out, right? If that's not a bottleneck, I don't know what is. It doesn't take $100 million of investment to get into one product that's going to come out, except within the very specific space of small molecule pharma type application, right? Especially when we're talking about materials products, chemical products, those ideas that we're investing in, we see bottlenecks there. And that's where Materic really likes to sit, is to break open that bottle and be able to give access to those technologies to come out. And I'm also excited to hear and engage with Elmarco that they're really investing in those kinds of incubation and innovation capabilities too at their side. And then just within, to give an example of that, within electrospinning and tissue engineering, if you Google Scholar electrospun and tissue, since 2020, you actually have about 20,000 publications that have come out in the academic space. We do not have close to 20,000 products, right? But those are all those ideas being generated that can really get empowered and developed by taking the step forwards to run off of an NS Lab type instrument. Eventually, the 500 units and the commercial units that Elmarco provides are really a great opportunity to step those from the academic space and into products that are really going to change people's lives for the better, and help improve health. We can see on these roll-to-roll instruments very similar fiber morphologies, and we've been able to show good translation between what we see with our academic collaborators that are doing things on needle-based systems to what we can get out on the roll-to-roll systems like the NS Lab. There is certainly a lift to go through that transition, but in almost all cases, if we have a starting specification of fiber diameter, porosity, area density, that we're able to get and tune a matching set of actual output physical properties from our material on the roll-to-roll systems like we could see in the lab-based systems, and usually much more consistently in a robust fashion, that we're getting good yields in that as well. And so this SEM image here in particular is a gelatin nonwoven material that we've produced. That's one of the materials we use in our line of biopapers, with fiber diameters around 400 nanometers average and pore size around 1.2 microns for that porosity. Okay, so we have roll-to-roll capabilities. Almost all the biopapers that we produce are actually on the needleless-based systems. Occasionally run into things where we might need a coaxial setup, that we also have capabilities in nozzle and needle-based systems as well. And one of the reasons we like electrospinning, especially for tissue engineering biopaper type applications, is even though the tool set is the same, we actually have a lot of different parameters that we can control in terms of inputs into the electrospinning process, that can give us very tunable outputs that we want in terms of the actual chemistry and composition of the fiber, fiber diameter, pore size, kind of how does water interact with that interface, what are some of the mechanical properties that we're trying to get to, and even things like degradation and alignment, which are actually all really critical. And I mentioned that there's those 20,000 electrospinning papers in tissue engineering that came out just in the last 3 years, right? There's a lot of unique attributes that you need for very specific cell lines, very specific tissues that you're trying to generate, that actually require that kind of tuning and control of a unique product for those cases. So talked about nanofibers. For those of you in the nanofiber space, that's all old news to you, but how does that and why does that apply to biology? It really comes down to the fact that cells don't simply sit immediately adjacent to each other inside of tissue, right? We think of it sometimes that way if we're not biologists, but the reality is that cells actually produce a microenvironment around themselves called an extracellular matrix. And that extracellular matrix is actually a three-dimensional non-woven nanofiber network, right? It's primarily collagen. Sometimes there's hydroxyapatite and minerals there if we're talking about bone structure. There's often supporting enzymes and proteins. But the biggest contributor of the volume of that space is actually collagen. And so it actually looks and feels like an electrospun material in terms of fiber diameters, pore size, and structure. And that's the environment within our own bodies that cells like to live in, right? It's what they generate around themselves. They'll often consume parts of that extracellular matrix and constantly be putting new collagen out to maintain that three-dimensional nanofiber network. But that's the home that cells like to live in. That extracellular matrix both provides structural and biochemical support for those cells. It maintains the geometric environment that that cell wants to live in. It often doesn't want to be a small ball of material. It actually wants to be stretched out in particular directions and geometries. And this cellular matrix is also giving anchor sites that the cell membrane will actually use specialty proteins called fibronectin to actually grab onto that cellular matrix, the extracellular matrix. So we have this non-woven set of fibers, and then the cell is actually attaching itself to those fibers in natural tissue, right? That environment actually also matters for gene expression and differentiation. So in a lot of tissue engineering, we're starting with induced pluripotent stem cells. And we're doing different things, either adding chemistry, adding hormones to that to provide a chemical biological environment, but also the stiffness, the binding sites available, the orientation of those binding sites actually directly influences if that pluripotent stem cell is going to become muscle tissue or bone tissue or something else, right? So that three-dimensional non-woven environment is actually really critical to how the cell chooses to express itself and how happy it is in the environment that it's in. And also within that, right, you actually want specific mechanical properties in terms of the stiffness of the individual fiber itself, as well as that matrix stiffness. and physical chemical properties, meaning what's the pore size, what's the frequency of those binding sites, what's their orientation and geometry, as well as is there supporting chemistry around that for that particular cell. So in research and in medicine, there's a number of reasons that people are very interested in these non-woven materials, really in simulating the extracellular three-dimensional environment that cells experience when they're in our own bodies, right? On the research side, it's been a major push to really move from what we call a 2D environment, which would be cells growing on the bottom of a well or a petri dish, to that three-dimensional environment. The reason that research is interested in that is that we run these cell cultures and we create these tissue cultures to then be able to test pharmaceutical products in the research industry. Maybe it's a cancer therapy, maybe it's a specific treatment for a disease case. But it turns out that a lot of those chemistries that we're testing in that 2D environment don't actually behave the same way when the cells are in their natural 3D environment. So there's a big set of experiments that look good in 2 dimensions for a particular cancer therapy we might be producing that end up failing when we move to that 3-dimensional environment in an animal study. Animal studies are really expensive, so if in the laboratory and the research side we can provide 3-dimensional scaffolds and 3-dimensional structures, then we can much more cheaply screen a number of those attempt therapies to be able to advance them more successfully into those animal and human trials. And then that's all on the research side of that tissue engineering. It's to create a tissue model that I can then apply chemistry to, see how that tissue model behaves, and have a better understanding of how that might work in an actual living creature. On the medicine side, right, so all of that research promotes individual pharmaceutical products that might come out. But on the actual medicine side, there's direct applications of these kinds of 3D tissue environments to patients. One is in regenerative capabilities, and there's even companies coming out with electrospun materials that have gotten through various levels of clinical trials. But two is to actually, and so that would be to take that material, put it on a wound, and allow healing of that. But then the dream is actually to be able to synthesize full tissue in lab, and that's really where tissue engineering comes in these 3D grafts, and then be able to actually transplant. The simplest of which is probably a skin graft at this point, right? And right now, a patient has to basically get donor skin from another portion of their body, often their leg, that's then taken on to heal a portion that might have been exposed to a burn or something like that. And then finally, we're seeing lots of interest in tissue engineering in actual cellular agriculture, meaning lab-grown meat or things that are avoiding traditional agricultural farms, basically taking the studies that have been done in the development of tissue engineering to actually produce synthetic, not synthetic meat, but lab-grown meat, right? So it's still using animal cells, but it's growing it in a laboratory so that we can be exclusively putting energy into that system to growing the meat, as opposed to raising whole heads of animal for slaughter. So this is our specific BioPaper technology that we produce. We actually make these materials in gelatin and PCL as retail products, but also are able to customize. So we've done different composite blends with that, different additives with that as well. And these are specifically for 3D scaffolds for tissue engineering. And this bottom picture here is basically showing an SEM picture of that 3D non-woven environment versus an actual SEM of the natural extracellular matrix, right? So visually, they actually link up in terms of how they look together. And we have this available in different insert sizes that you can put into traditional well plates that you might be doing cell studies on. And you can actually see, and there's a large body of papers that are looking at cell and cell proliferation onto these types of synthetic extracellular matrices that really show that there's good proliferation over time periods. And so anything that's green means that it's alive in these kinds of images. And so that cell viability is really there and in good shape off these types of scaffolds. In terms of what we've done with those specific to gelatin off of our Elmarco systems, we have them available in those inserts. These are anywhere from about, I'd say, like a 5 mm diameter to a 2 cm diameter, but we're taking sheets of non-wovens that we produce and cutting them and putting them into these inserts for use in those cellular tissue applications, but we can also do things that are larger screens. So we can have as spun material like this on the left. These bottom two pictures are for typically this would go into well plates for people who are doing studies within a research or a laboratory setting, but we have customers that want larger sets of 3D scaffolds, and we can do these different framing processes to basically cut the electrospun material out to have something more like a 15 by 10 centimeter area that they would use for a bioreactor or an individual piece of tissue that they were trying to generate. One of the reasons we're doing these screens or these insert type processes is we are often doing a secondary process after electrospinning before that material is going into that tissue step. These two cases, we're talking about different cross-linking methods. If you take electrospun gelatin and you just put it into cellular media, it's going to dissolve right away, right? It's already a soluble polymer, and then it's a nanofiber, so you've got all that surface area that's available. It's going to disappear almost instantaneously. So we cross-link that either using Genipin, EDC NHS, GelMA type structures to basically lock that nanofiber structure in place. And you can actually tune the degradation time of our synthetic extracellular matrix by tuning the amount of cross-linking. So we can get it to last 5 days in the environment where maybe the cell or the tissue has started to create its own network around it, or we can get it to last 90 days in tissue that way. Okay, so then this gets more into specifics of tunable properties that you can start to do and control, and what that means in terms of outcomes for individual cells. And so this you can tune just based off of cross-linking. What's the stiffness of those fibers, right? The more I cross-link, the stiffer I'm going to be. And then that directly has an outcome for the same geometric structure for some of these stem cells into how they're going to differentiate. So a softer substrate under the same chemical and otherwise physical environment could result in a neuronal cell coming from that stem cell at around 1 kilopascal stiffness, right? If I move to a more stiff material just by cross-linking more, going to the 10 kilopascal stiffnesses, you're going to actually induce muscle tissue out of it. And then if you go all the way to a very rigid environment, 100 kilopascals, you'll start to get bone-like cells that grow out of that, right? So those cross-linking, those chemical work that you do to give rigidity to that scaffold can have direct impact on the tissues that are coming out. And then also you can dope your electrospun material with drugs that could help with vascularization or other kinds of growth factors that are also promoting either differentiation or proliferation of the cells. So I've kind of broken up the rest of the talk into thinking about what are electrospinning parameters and outputs that we can think about controlling, and where does that parallel into the biology, right? So the first thing I've broken up is that tuning of the physical structure or morphology itself. That means your fiber diameter, your pore size, your alignment, or also if you need a hierarchical structure or a mesostructure. So your fiber diameter matters because there are actually desirable diameters for cells to be able to sample to get that fibronectin to grab on and latch on to those adhesion sites, as well as desirable aspect ratios. So if you have fiber diameters that are too small and then fibers are stacked too compactly together, it might start to look again like a flat sheet of material to the cell, and it'll start to behave in a 2D fashion, right? So there's tunable diameter ranges that you want, often in the few hundred nanometers to single micron range. But you'll see different kinds of outcomes from your cells based off of that. That's tunable based off of your solids loading, your conductivity, as well as the actual voltage that you're applying to the electrospun system. Your pore size, right? How dense those fibers are actually intertangled, directly relates to cellular infiltration. So certain pore sizes, cells are often, let's say, in the 10 to 20 micron size range. They can move things out of the way, but there are certain pockets that they might be happy in, and certain other pockets that are maybe too small. It's like a Goldilocks kind of scenario where you want to sit in the right size to have the right environment. It also matters for pore size because all of these scaffolds have to be able to perfuse, meaning that when I'm applying new nutrients and media to the tissue that I'm growing in lab, it has to be able to get to the cell itself. So it has to be able to get through the scaffold to the cell. The cell's consuming and metabolizing those products and putting out waste products that also have to get out from the cells and out from the tissue to the rest of the space. So by having this three-dimensional scaffold, you're able to allow that perfusion to go in and out of that tissue structure, just like in the case of air filtration, right? Nanofibers are great because you can have a lot of surface area, but you can get a lot of air through them at the same time, right? So that same physical concept of that structure is very valuable in tissue engineering. Alignment is very important, especially for certain tissues types. And that's where that rotating drum or collection matters, where if I've got a randomly aligned or oriented tissue versus an aligned, that can make a difference both in differentiation as well as if I have scar tissue that forms. So cells that are aligned tend to be in a nice uniform oriented structure. And then even mesostructure, so sometimes cells in natural tissue have these honeycomb-like structures. This is electrospun material just by changing the grounding electrode geometry through Smith-Rouse group at Michigan Technological University. In terms of mechanical properties, we talked about that a little bit, but in terms of electrospinning, you can choose your base polymer to start. So among Biopaper materials that we've made, we've done gelatin, PCL, PVA. Gelatin's a lot softer than PCL, so that's a primary thing to be able to change that stiffness out of. So your polymer choice is a great thing to start for picking that mechanical property. You can do composites of that. You can do gelatin PCL mixtures, PCL PVA mixtures, and other things to try to tune in between those different just base polymer properties. And like I said before, you can also cross-link to change stiffness, rigidity, as well as degradation time of that material. And then compositing, we can do both in the spinning process, so we've included APIs or other ceramic components in the spinning process that might change that mechanical property. Or you can do things post-process to use an ink or an additive of some kind that would just cover the surface morphology. This is actually a paper that was done on our biopapers themselves, gelatin cross-linked with different cross-linking techniques as well as different concentrations of cross-linking that we showed cell proliferation on, as well as tunability of how long the original electrospun matrix was in place versus then the final natural biological matrix eventually took over and took shape in those materials.
I can speak about this. Hi, guys. So just going off of Steve's discussion about additives in nanofibers and mechanical properties, a lot of work has been done to kind of tease out those individual qualities of nanofibers to really determine what's contributing to these cellular outcomes. A lot of the times you turn one knob on your electrospinning process and you end up getting a lot of downstream effects. So I'll kind of go into this one case study where authors were actually able to decouple the effect of stiffness and roughness on nanofiber composites using multi-walled carbon nanotubes. So by increasing the percentage of multi-walled carbon nanotubes in a PCL nanofiber, they were actually able to pinpoint exact concentrations where stiffness and roughness were maximized and minimized, and were thus able to arrive at different phenotypical changes in MSCs that they were mesenchymal stem cells that they were differentiating into bone tissue. So this is a really interesting study because you could see that in the middle panel here, the higher stiffness nanofibers cause this sort of rounder morphology, while the higher roughness fibers cause a more elongated morphology, and that has these downstream effects on bone biomarkers, osteocalcin and osteopontin. So this allows folks who are actually using nanofibers in applied settings to pick and choose their roughness and stiffness according to what they want. Osteopontin is a late stage bone maturation marker, whereas osteocalcin is an earlier stage bone maturation marker, which could result in an increased recruitment of osteoclasts and immune response that you may not want to have in an implantable dressing or wound healing or bone healing scenario. So you could go to this data as a researcher and say, okay, I want something that has a carbon nanotube concentration of 1% because I want that osteocalcin, but I don't want that osteopontin, or the opposite. In terms of the nanofiber alignment and how this can affect cell morphology. So I know there was a question about how geometry could be used to organize or align cell structure. So this is a great example. On the left, we see how nanofiber alignment affects the outcome of neuronal cell differentiation with different neuronal markers upregulated in the presence of either aligned or random fibers, respectively. I'm sorry, there wasn't a key here, but the gray is actually random and the white is aligned. So you can see that certain biomarkers are more highly upregulated with certain alignment. And you can also see how that changes according to different nanofiber size. On the right, we see how PANI nanofibers were used to alter the orientation of myofibers and thus affect the actual maturation of those myofibers. So with aligned nanofibers, which more accurately mimic the structure of muscle tissue, you were able to see much greater myotube formation. You get next. So, why is this happening? I think it is an enormous research thrust by nanofiber scientists and tissue engineers to actually decouple and figure out why these are happening. And Steve kind of went into how cells communicate in their microenvironment, but I think this paper does a really good job. So the Bowers, this is from the Brown lab at Penn State, goes into exactly how the nanofiber thickness and alignment is actually responsible for controlling the focal adhesions of intercellular communication. And I won't go into this entire image, but you can see that it's not just the alignment that controls cellular outcomes. You actually have the thickness or the thinness and the alignment both working sort of in symbiosis to affect those outcomes. And you can see in the far right image that in the second image over here, where you have increased B-catenin or Wnt signaling, which is actually causing the increase in myogenesis. Sorry, that's the third image where you have thick but also aligned nanofibers over there. So because you're basically increasing the amount of Wnt signaling caused by the B-catenin focal adhesion points, you're increasing that myogenic regulation. Another really simple part of the equation is mechanical properties. In aligned fibers, you are inherently going to have stronger nanofiber mats, and those will also affect cellular outcome. But of course, that is always dependent on, are your cells growing along your fibers, or are they growing above your fibers? And mechanical properties will have more or less of an important impact on that. You can go next. So, I mean, nanofibers can be really interesting drug delivery vehicles. And this is a paper where they actually used gradient structures in nanotubes to create these sort of vascular grafts loaded with growth factors to control the growth and differentiation of in vivo conditions. So, if you want to stimulate growth and then have that material degraded until you're stimulating differentiation, that's something that you can achieve with these protein-loaded nanofiber grafts. You can go next. And I just wanted to go over, this kind of wellspring of in vivo conditions and in vivo tests that recently have been pioneered by various labs using nanofibers. So on the upper left, cartilage was replaced with nanofiber matrices. And this kind of took advantage of the porosity of nanofiber mats that allowed for diffusion of nutrients and oxygen through tissue, which is something that you can't achieve with hydrogels. Going to the right panel, this went over showing how nanofibers could be loaded with bioactive compounds in wound dressing, and was able to achieve much better outcomes than just 2D treatments because of the increased surface area and the perfusability of those nanofiber mats. On the lower left, this kind of took advantage of the fact that you can, like Steve was saying before, really customize the geometry of your nanofiber mat by spraying onto electrodes that are of different shapes. So this could be really good for solid bone tissue engineering. And on the lower right, we have an example of nanofibers being used for cardiac tissue engineering, where the piezoelectric properties of PVDF are harnessed to enhance the electrical conductivity of these patches. So like Steve was saying, nanofibers have these really unique properties where dipoles can align and create the electrical impulse that you see from piezoelectric materials, and these can be harnessed for tissue engineering applications. And so kind of my bread and butter and really interesting application of nanofibers is applications to cultivated meat. So this is, of course, a field in which you are trying to grow meat without the use of an animal in tissue culture, and sort of two pioneering studies in this field using nanofibers, where on the left, Luke McQueen up at Harvard used a rotary jet electrospinning process whereby nanofibers were basically spun in your classical cotton candy machine, whipping up those fibers using nothing but centrifugal force, and was able to grow rabbit satellite cells on these fibers, and they grew really well and aligned with those individual fibers. And currently, this company is actually very much present in Boston, Massachusetts, where I live now, and selling their product at restaurants around the city without the cells. So they're just selling sort of their fibrous material. And on the right was an interesting production of an actual fully formed piece of meat using nanofiber technology, whereby cellulose acetate nanofibers were created and stacked and seeded repeatedly to actually create what is a whole cut piece of meat that they were actually able to cook on a frying pan in that panel. So a lot of the benefits of nanofibers being sort of tissue-like and tissue responsive and able to support the cell growth and differentiation is really important to this field.
Great, thanks, James. And I think with that field as well, it's something where if it has to actually go into production, you need large quantities, right? When we're talking about tissue engineering for medicine, it's something where it's always going to be pretty boutique and individualized, right? We're going to create an individual tissue for an individual patient that has specific needs. And so I think that always sits in a particular scale and probably local to some hospital system, right, that's going to be treating that patient. But in the cellular agriculture space, we are talking about an industry that you actually need to get to large commercial production for it to be like fully viable. So it's pretty interesting to see what we've been developing for medicine also coming into the food space and the nutrition space as well. But then to summarize that, right, we know that cells naturally develop actually in a nanofiber matrix environment, and that electrospinning provides us really a scalable means of synthetically producing a matrix to then get cells to develop and grow in these kinds of tissue-like environments, which allows them to eventually take on their own tissue-like nature, right? When we're starting with loose cells that we've extracted or grown in culture, they can now have the right mechanical, physical, chemical, biological environment to become a full-fledged piece of tissue. And we also have this tunability in your fiber size, geometry, mechanical properties, and signaling by using additives to really have those unique properties of the scaffold we're generating, and then to get those very specialized environments that different tissue types would like or want. And then the last slide is just, oops, talking a little bit about our team at Materic. We've got those experts in electrospinning as well as in a number of those other supportive areas. James used to be part of that team. That's why his background in this picture is the same. But we've seen him go on to a PhD program at Tufts now, where he's specifically working on cellular agriculture systems and biomanufacturing for protein and food sources. And then a lot of what we talked about in our talk today, we actually have a review article publication on that was just published last year. But some of the things that we also cited were published after that review article came out. So again, this is a very active development area, both in academia and in industry. So there's a few things that we tried to include today that are even more recent than our review article a year ago. And with that, we're happy to take any questions. This is my contact information if you need to reach out, or you can go to our website if you're interested in sampling any of we've got different gelatin PCL biopapers. This particular one was cross-linked with EDC NHS and also has a collagen coating to kind of give more binding sites for adhesion than just the gelatin itself. But yeah, James and I thank you for joining and for our talk, and we're happy to take any questions.
Yeah. Thanks, Stephen. Thanks, James. Very interesting talk. First question for me is, how does the meat taste out of the lab?
Yeah, I can definitely answer that. We actually had our first cultivated meat tasting in Boston last week, and it was great. We had a company called Wild Type, which makes lab-grown sashimi, come and do a tasting. And yeah, I mean, there's always things they could, one could work on, that they could work on. I would say the taste was almost there. I was surprised by how actually fishy it tasted, but texture is always something that could be worked on. And I'm really into sushi. I'm really into like sort of the chewiness of it. So I think again, it's something that it all comes down to fibers and texturizing meat with materials.
Mm-hmm. Okay, thank you. We have quite some questions in Q&A session, but before that, I have my question, which is related to sterilization of the nanofiber mats before you let the cells grow. So how could you describe, how do you do that? Because I know that some people are using sterilization by ethylene oxide. So is that your method or using some different method, and is actually needed before you let the cell doing the job on the nanofiber mat?
Yeah, so I can talk generically and then let James talk about tissue specific, but the sterilization technique is often specific to the polymer set that you're working with in that 3D scaffold. So for gelatin and PCL, we're fortunate in that we can basically just do an ethanol soak to sterilize, and we would do that within a biohood itself, which maintains a sterile environment otherwise for the air circulating through, and that works. But yeah, the ETOH or other things, depending on the polymer set. So you have to check what polymer you're using, that your sterilization process isn't going to damage or affect the structure in any way, but then you can work from there. But for tissue engineering, you really always need to sterilize before doing the cell work. But then James can talk from a more practical experience as well.
Yeah, I think with ethanol treatment, there's always the risk of, depending on what polymer you're using, it'll get dissolved or ethanol will get trapped in there. And I think as these technologies scale, we're really going to see more just in mass ETO sterilization, which hasn't I haven't seen it affect nanofiber integrity. So that's kind of a more industrially applicable approach to sterilization that we could foresee doing. Yeah.
All right, thank you.
There's a few questions about loading it with bioactive agents or with APIs or anything like that. How does that affect the porosity and how you're working to differentiate and proliferate these cells?
Yeah,
Yes.
sorry
James,
about that.
you go first.
No, I do see a lot of questions about bioactive loading, and I know, like in our experience at Materic, it is very particular to what you are actually trying to load it with. There are some things that have a very seamless integration into materials where it's a small API at a small concentration and there's no difference, and then there are things like nanoparticles that could have an enormous impact on your spinning qualities. So it really is a case-by-case basis. Once your material is loaded, there's a couple of questions about controlling release of drugs. And I think that really has to do with a multi-factor effects from the porosity, the nanofiber material, the nanofiber diameter, something that you would really need to tune with your own with whatever you're trying to do with it. The one thing that you can do with nanofibers is create core sheath double layered nanofibers, which you can do with instruments like the Invivo, where you could actually have biphasic release of your drug or your active product. So
yeah, I think so. To James's point, we've added a number of those different ingredients, either as inorganic ingredients like nanoparticles, or organic nanoparticles as well, right? So something that might be a drug-loaded capsule or a particle of some kind that's getting incorporated as basically co-spinning. And yeah, depending on the drug type, the molecule type, it may or may not influence the conductivity of the solution, the surface tension of the solution, or in those nanoparticle cases, sometimes they're actually a focal point for the electric field to concentrate around. And so it will change your kind of density of Taylor cones and things like that. So you can do it. And depending on what you're working with, you can figure it out. But we've definitely spun mats of pretty high quality in terms of there isn't fiber shedding or large issues with uniformity using a number of those things. I'd say the one thing that we try to be cautious of is if you are using a protein or an enzyme as a specific signal or additive to your system, those can be very vulnerable to pH or different solvents to denaturing. So for a number of those things, like we've got the collagen, or James was talking about, you can put materials on the outside or core shell fashion. Sometimes it's easier to make the nanofiber structure than do a wet process to layer that next outer shell on the material, because you're then using solvents or environments that aren't going to risk denaturing of those ingredients, right? So collagen would denature in a number of the different solvents that we use in electrospinning, but we just were able to do kind of a soaking bath process afterwards. that's able to keep the collagen within its triple helix kind of native structure without influencing it. So I think that's a major consideration too. If the ingredient you want to do is a macromolecule, a protein, or an enzyme, you have to be cautious of the environment you're subjecting that protein to make sure it doesn't denature and you're just putting a set of amino acids randomly on it and not the right tertiary structure that you need to have the activity you're looking for.
Maybe back to the general structure of nanofibers. So can you generalize like what is better if you have thicker fibers or bigger pore sizes? So can you like do it comparison between each cells, or is it pretty much relevant to each cell that they prefer? I don't know what pore size.
Yeah, I mean, I'd say there has been a lot of investigation into porosity. I think porosity becomes the most important when you're trying to create perfused 3D structures, where you're actually trying to get nutrients into a core where your cells might be growing. Unless your cells are actually growing inside of the pores, they are generally not affected by the pore size as much as they are about the nanofiber qualities, because they're growing along the nanofiber. Now you have a lot of you have a lot of cases where the cell's just growing over the whole nanofiber mat if you have very fine nanofibers and they're not actually growing along the nanofiber, in which case, again, the pore size really is impacting their ability to uptake nutrients in the media and have those nutrients perfused. Impacts of the pore size on the actual focal adhesion and differentiation outcomes is kind of a difficult one to really pinpoint, because the pores are just a lack of material. So how do you study sort of a lack of material impacting the nanofibers as opposed to the actual morphology of the nanofibers on the cell itself? But yeah.
We're over just a few minutes, but we're going to continue for about 3 more minutes. So I'm going to take a couple more questions. We've been kind of talking more about the material itself, like how the cells work and the material you're producing. There is an electrospinning question asking about how do you control the morphology of the fiber and probably this porosity and everything by controlling the gap, the gap distance, maybe the spinning to the spinning to collector gap, and those types of things. What are the major things that you do, major levers that you do to control these things?
Yeah, so I think controlling the collector is the biggest thing you can do to affect alignment. And then with porosity, it's solvent changes and it's post-processing, like leaching out. So basically spinning with a sacrificial polymer that you can kind of solubilize out with water to get rid of and increase the porosity. There's not a lot of sort of in situ as you're electrospinning approaches to alignment. Maybe Steve can speak more to that since he's on the cutting edge, but yeah, I haven't seen a lot. Yeah.
Yeah. So I think that the control, so with a particular GSM or weight area basis, right, we have some controls in the tool as it is in terms of your electric field strength, which is what you're putting on versus that distance, right? If you kind of have enough electric field strength to generate the fibers, but it's not quite as high a driving force to push them into the substrate, you can end up with the same area density mat, but a larger overall thickness of the substrate that you're working with, right? So we can change the actual volumetric density by plus or minus kind of 60 or 70% relative to maybe like a middle line density that we've seen just by tuning that spacing of the electrodes, even spacing of the grounding wire, in our case for our NS Lab instrument, versus where our collector substrate is, that we can tune that density to a certain extent. James is referring to another technique we need if we need to get a much, much loftier, much more porous material. We might co-spin gelatin with PEO, and the PEO is soluble in ethanol, but the gelatin is not, right? And so we actually co-spin with a sacrificial polymer, and we might cross-link the gelatin in place and then actually dissolve away the sacrificial polymer, and then you can end up with a really lofty material. I think those are things that we do in these specialized cases that for like a commercial nylon filter you never think about, because it's so much extra processing for something that's a commodity product. But those are kind of techniques that you can really do in these specialty tissue engineering spaces that the price is in the actual cells and then the culturing of the cells and not that substrate that goes into it. So you can get really creative in processes to try to change density and do things like that as well.
Well, thank you guys. I think we probably need to wrap it up, but there's a lot of questions that didn't get answered, but we will send those, we will collect those and we will send them to whoever makes sense to answer them. And we'll make sure to get those answered as soon as we can. I just want to remind you guys to be on the lookout for the next series that we would be sending out information about, and also an evaluation. But Stephen and Steve and James, please, thank you for coming, and thank you to the audience for being here. It was a very interesting talk, and we could probably go on for another hour. So, but I appreciate it, and I appreciate everybody that attended today. So.
Yeah.
So webinar session is over. Like for now, for a couple of months, we are taking a break, but it doesn't mean that we stop communicating with you. So please, whatever you have in your mind related to nanofiber application, come back to us anytime, basically, to reach us out on our email addresses or Elmarco website.
Okay.
Thank you. Okay, bye-bye.
Thank you all. Take care.